Scientists can now sequence 3 billion bases of DNA in a day's time. The first human genome took several years to complete just a decade ago.
It's very hard to keep up. Now you get out so much data that it costs almost nothing to sequence DNA. Most of the cost comes into preparing samples and analyzing the results.
As speed increases, so does demand. Jim Lund is the senior scientist at ACGT Incorporated, which sequences DNA. Their clients range from academic research institutions to biotech companies.
The sequencing machine is a fairly simple technology.
It's become so simple that genome sequencing is now less costly than analyzing the data it produces. Sanger sequencing has been used since the late 70s.
Sanger sequencing costs basically as much as a sandwich. So Sanger sequencing will give you 800 or 1,000 base pairs, starting at a point that you know about. So you'd say, starting at the beginning of this gene, give me 1,000 base pairs.
Sanger was the standard until about 5 years ago, when next-generation sequencing became more accessible. If Sanger is like spearfishing, next-gen casts a much wider net.
Next-generation sequencing is you give it a bacterial genome, let's say 5 million base pairs, and you sequence all of it 100 times over. And then you say, well, okay, let's look through that sequence for the piece I want, and then we'll look at that piece. Now you can just take the DNA, you can get it sequenced in a couple weeks, and then you can say, okay, well, now I've got the sequence, let's compare it to a benign organism or to a known organism and see what's different, and just pull out the interesting parts.
The process is now easier than ever. A swab from the inside of your cheek contains 1,000 times more DNA than Lund says he needs to sequence your entire genome.
It's DNA in tubes like this, and usually only the little drop of water at the bottom. It's going to be very exciting as people it becomes cheap enough for people to use it to diagnose disease and to help human health.
Lund says he's excited about what lies ahead for the field, even if its future is uncertain.
We can sequence your genome now. We can't tell you a lot about yourself from that. There isn't a lot you can do with that information today, but it's going to be more and more informative as time goes on. What happens with that? Well, that's something society is going to have to figure out. It's not clear what we're going to need to do, what we can do, and what we want to do.
For Medill Reports, this is Matt Rhodes and Corinne Chin.